Welcome to Let's Talk Oral Health by Gum, your source for conversations on oral health and beyond. My name is Rachel Shaw, and I'm here with my co-host, Martijn Verhulst. Hi, Martijn.
Hello.
How are you?
I'm very well.
For today's episode, we are very honored to welcome an expert on oral microbiome. And with him, we are going to dive into the fascinating world of microbes and their interactions between themselves, but also with the mouth. I'm talking about Professor Wim Teughels. Hello, Wim.
Hi, Rachel and Martijn.
Hello.
Thank you for having me here.
Thank you for accepting our invitation to come to Sunstar headquarters in Switzerland. We are very happy to have you with us.
It's my pleasure.
We are very eager to start the conversation on oral microbiome with you. But before we do that, let's start by introducing you a little bit to our audience. So you are a professor of periodontology at the University of Leuven in Belgium. Mm-hmm. Can you maybe tell us a bit more about your career, your research area, and what you do over there?
From an educational point of view, I'm a clinician, I'm a dentist who actually got interested in oral microbiology during his dental training. And it's a little bit of a strange story, but I actually got interested in oral microbiology by being lazy. And the story behind that is that in my, I think, third or my fourth year of dental school, I need to write up a thesis, like everybody, a master thesis. And I didn't really feel like writing too much and doing an extensive literature research. But I had a friend who was doing a PhD in oral microbiology. So I thought if I do something in that field, he will give me some data and I can write an easy master thesis. So I applied at the perio department to connect up and to work together with my friends. And I was accepted, but they connected me to another PhD student who basically pushed me to work day and night in microbial culturing, one plate after the other. And I fell in love with these bacteria. So I ended up in oral microbiology. So the rest is history, doing the normal postgraduate oral microbiology PhD. During my PhD, I came into connection with Mike Newman from UCLA and basically was now the head of the Carranza's textbook. We connected well. We started to chat about microbial interactions. This led to the probiotics field and my career started up. So now I'm focusing primarily, not only, but primarily on microbial modulation and modulation of the oral ecology.
And you do research in this field of microbiology, but you also still treat patients, if I'm not mistaken, right?
I'm still very, I think almost 40% of my time is still patient treatment. And I really see this as an advantage. In the old days, I felt always bad because you have these really big basic scientists that really can go dive into the molecular microbiology, which I couldn't understand, but I was also not a real clinician. Now, so many years later, I really see it as an advantage because I can talk to the both sides.
Sure.
And I can easily bring experiments of things that we notice in the lab to my patients really fast, but also treatments or changes or experiences of my patients, I can really easily bring them back to my lab and see what is the reason behind that and how we can use this. I really feel myself more as a translational research, bridging the two fields, and I'm very happy with that. I would not want to have anything else. So
it's not a very common combination, as you said. Usually they're either very fundamental basic researchers, or you have the very much clinical practice focused type of.
That is the fun part, because I can actually use that outside of the oral microbiology field. So I'm also involved in some environmental probiotic, prebiotic modulation projects, and also there my contribution to the project is often the translation of things to real life settings and back. So that's a thing I love to do.
Yeah.
But we are definitely going to tackle into these two expertise of yours during our conversations, because we are always trying to indeed bring scientific information to our audience, but try to also have an application, a pragmatic application to that, since our audience is mainly oral care professionals. So maybe let's start to dive now directly into the topic. And I would like to start with the shift that we have encountered in the past years from the concept of periopathogens or chirogenic bacteria towards a balance of the ecosystem. Can you maybe set the stage for us and explain this paradigm shift that happened?
So when I still was a student, what we often did was we looked for specific pathogens. We knew, okay, periodontal disease is related to these and these and these pathogens. For tooth decay, we have the typical strep nutans, strep sobrinus, and we were focusing on eliminating these pathogens. So often when I still was a student, we were taking microbiological samples to see what was there. At the end of therapy, we were checking whether or not they were still there or not. And often it was a disappointment because they were still there. Now, over the years, logically, we see this shift and a change in our understanding that actually almost every human being is being colonized by these pathogens. They are everywhere there. But we start to understand that there is this whole bunch of bacteria that we were not interested in the 2000 up to 2010 that actually play a crucial role in oral health. So the importance of something which we call commensal / maybe beneficial bacteria became way more clear. And then pioneers like Philip March came up with the concept that, look, it is not these pathogens that are of importance, but it's the whole microbial community. It's not only about their numbers, but it's also how they interact with each other, how they interact with the immune system, how their metabolic activities, which virulence factors that they produce. And this is all regulated very tightly between these groups of bacteria. This made that by 2015, something like that, we actually left the concept of looking at specific pathogens. We know that the old pathogens that we know basically from the 60s, the 70s, that they are in a way a little bit of a marker organisms for how what we call now dysbiotic your biofilm is, or how bad your biofilm is, or good our biofilm is, but it's not a treatment target anymore. They can give you an idea, an indication, but it's not a treatment target. So,
sorry to interrupt that, but what at that point in 2015, what triggered that change of perspective? Because there had always been this belief, at least for a few decades, that these were the pathogens that you need to eliminate. So what exactly was the new insight at that time that you know?
I think it was two things. On one hand, it was the Philip March ecological plaque hypothesis that came up around that time, where you really had to, both for tooth decay and for periodontal disease, the shifts. That was one thing. Then we had also the keystone theory, which came up from George Hagian, sorry for the name, Hagian-Delitzsch. And in a way, they matched to each other. This is also the period where we started to have insights with the whole genome sequencing.
Oh, sure.
So we started to realize that there is way more than just these 10, 15 bacteria where we have been focusing on. And that even in healthy situations, they were still there. So we couldn't find one or two or three typical pathogens, what you would normally find in an infectious disease. But we saw that things changed when you went from health to disease and back. And this was basically the basis of the whole concept, that maybe we should go away from the classical approach of killing all the bacteria in the oral cavity to shifting them, because probably it's impossible to kill them. And then a second angle came with the recognition, the understanding that these beneficial, I will call them beneficial bacteria, that they actually play a very important role in keeping these balances straight, so that they were suppressing the outgrowth of pathogens, but that these beneficial bacteria were also very important in modulating the host response, modulating inflammatory responses. And this leads actually to the current concept of microbial balances and trying to create stable ecologies rather than eliminating bacteria.
But then when you talk about balance, you are then mentioning eubiosis, to which we have to oppose, you already mentioned it, dysbiosis. So can you detail a bit these two notions and what happens when dysbiosis strikes?
Eubiosis and dysbiosis are two terms that you will hear more and more also in the future. So when we are talking about eubiosis, then we are talking about a biofilm, a microbial ecology, which is in balance with health. So most often this relates to high numbers of beneficial bacteria, lower numbers of so-called pathobionts, pathogenic bacteria, and something which induces a low inflammatory response. This is eubiosis, something which relates to oral health, periodontal health.
Yeah.
When we talk about dysbiosis, then we're talking about a situation where you have an outgrowth of certain groups of bacteria, most of the time bacteria that are recognized to having a lot of virulence factors that are able to trigger the inflammatory response. So dysbiosis is often also connected towards an increased inflammatory response, increased bleeding on probing, increased virulence. So these are the two extremes. Now, what happens is due to environmental changes, an ecology can go from an eubiotic state to a dysbiotic state. Now, one concept is still important there, that is resilience. So eubiosis is, although it's a healthy situation, it's actually a very stable situation. This ecology really can withstand a lot of pressures from outside and still remain in a eubiotic situation. Now, when you went from eubiosis to dysbiosis, to a pathogenic state, unfortunately, this stability, this resilience also establishes. So also the dysbiotic state can be a very stable situation. And this is actually hampering treatment, because if treatment is not really focused well, immediately, although you can induce a little bit of the shift in dysbiosis toward eubiosis, the resilience will bring it back to a dysbiotic change. And this makes that maybe in the future we have to work on these biofilms in other ways in order to bypass this resilience, to bring it in a different state, probably a eubiotic state, but again with sufficient amount of resilience.
Going back to this eubiosis, dysbiosis, trying to think about it from I would say like a temporal way, in the way how it associates with health and disease. Of course, I can imagine dysbiosis is associated with disease, periodontal disease, caries, whereas eubiosis is considered with a stable, healthy situation. Does a dysbiosis always precede a disease? Or it's a bit of a tricky question, like the chicken and the egg. You know, what comes first? How do those two situations, so dysbiosis to diseased state, how do they relate in the temporal basis?
Based on the most recent data, you can look at it from different perspectives. So, for instance, for dysbiosis, I could say, well, Martijn, what's first there? Are the pathogenic component of your biofilm growing out first, and is that then reducing the beneficial component? Or is the pathogenic component growing out because you're losing the beneficial component? Right. On the other hand, you could say with inflammation, do you first get inflammation, which improves the dysbiosis, or do you get first dysbiosis and then you get inflammation?
Yeah.
Probably it's something that runs a little bit in
parallel.
parallel.
Right.
But based on very recent data that I saw, some molecular biologists seem to tend that you can already see dysbiotic states before there are clinical signs of disease.
No.
It might be that the dysbiosis triggered to something precedes the clinical signs.
Yeah.
That is very sure.
Mm.
If it precedes inflammation, I'm not sure. I think it's really a coordinated, balanced interpretation.
Yeah. We always refer to this like this vicious cycle. But then even in a cycle, of course, it's the question where does it start and how do you break that cycle?
Yeah.
And that's always. I mean, as you said, it's a tricky question, but it's an interesting concept, I think.
Yeah, it is.
But I would like to come back to something you said about working on resilience, basically to make it work in our favor. What are the factors that could affect the resilience that we could work on?
Resilience is one of these newer concepts. So the factors are still a little bit always with a question mark. But what for sure is in favor of resilience is a good oral hygiene. Basically meaning a good plaque control, a good biofilm control. Don't let it become too much. Although I admit that I have for sure seen patients that have a huge amount of plaque, have actually hardly any biofilm control, and are extremely resilient towards periodontal disease and tooth decay. They exist. This is the minority of the people.
Yes.
On the other hand, it has a lot to do also with inflammation. I think if you can keep a low grade of inflammation, low grades of inflammation will actually will help in resilience. Or the other way around, if there is a lot of inflammation, the chance that you stay in a resilient eubiosis state is very low.
You mean a local inflammation or a systemic inflammation, or both?
Actually, both for me. In my perspective, based on what I've read and seen in the literature and in studies we did, I think that we don't have to underestimate the systemic inflammatory pathway. I think this is extremely underestimated. The local is very easy, that we understand.
Yeah.
I mean, you have a gingival sulcus, a periodontal pocket, there is bleeding, there is a lot of serum, so there is proteins. These groups of pathogenic bacteria are often proteolytic, so they can use it. And these proteins can inhibit, for instance, the peroxides that are in the biofilm that keep the pathogens under control. It becomes a different story with systemic inflammation because it is not that extreme. You often don't see it, but it's a low grade of inflammation that is actually triggering the immune cells to be hyper responsive. So you get a different type of macrophages, for instance, just because there is systemic inflammation. It's something you don't see immediately clinically. You don't see extreme redness or whatever, but it makes the patients more susceptible to going into a dysbiotic state.
Yeah. So your inflammatory response is more exaggerated,
indeed,
than it would have to be.
That's why I really think that we have been talking for a long time about the oral health systemic health condition, which is something which is really well established, where we have really evidence that it is there, how it works, for which diseases it's important. But now you feel that within at least at the moment in the scientific community, we are more thinking in the other way. I would call it the gut microbiome, oral microbiome connection, where we are wondering to which extent the gut microbiome and the systemic inflammation that it is causing. We all know this from all sorts of irritable bowel syndrome, Crohn's disease, how important this is. So we are now wondering how much maybe dysbiosis or eubiosis in the gut microbiome, which also exists there, is actually influencing the oral microbiome.
Sure.
And I'm honestly, personally, I'm a big believer that it is a very underestimated pathway that we are looking at. And it might be very surprising how strongly the gut microbiome is actually influencing the oral microbiome without knowing.
Communication line in the gum-gut axis, basically.
I mean, if you think about how we have data these days, how the gut microbiome can influence, for instance, depression. So there is a gut-brain axis, very clearly shown, very nice research, both in animal research, in clinical trials. I mean, if the gut microbiome can do that, then it for sure should have something on.
Especially because they're part of the same tract.
Yeah,
they're part of the GI tract. Yeah.
And you have a constant influence. I mean, the raw bacteria coming into the gut might be a little bit more difficult for the gut bacteria to come into the oral cavity, but in theory, with the new translocation mechanisms that I see being understood now in science, how these bacteria can travel from one remote site in the human body to the other remote site, then I would not be surprised that it would also go into the other direction.
Yeah, maybe it's not even the translocation itself, but it's through other mediators that we maybe do not even measure yet.
No, for sure. Yeah, but for sure the inflammatory pathways and the immune modulation is a very important one. It's not all about the bacteria; it's the combination between the host and the bacteria, and the immune system is there. They're a very important aspect of.
You mentioned one of the factors, just to come back to the resilience, to how to act on it. You mentioned the oral biofilm, basically how to control the oral hygiene that allows then to work on the oral biofilm. Can you maybe talk a bit more on maybe when you have an accumulation of a biofilm, you have communication probably between the bacteria, like some sort of influence on the roles of each other. So how can you maybe detail that to us to understand really the role of the biofilm in resilience?
The biofilm allows a very close communication between different sorts of bacteria, and it's very well known that a biofilm is just not a bunch of bacteria that are by coincidence next to each other. So they actually build intensive networks, collaborative networks, where they use each other's metabolic pathways. Now, this means that the whole biofilm process is biofilm buildup process is very specific. But this also means that if you disturb this process, you actually can slow down the transition of biofilms towards going into a disease state, because each time these biofilms or the bacteria in these biofilms need to reestablish these metabolic networks, and that takes time. The fun part there is because we start to realize that there are these metabolic networks is also points of engagement. I mean, if we know which metabolic networks are working, we could interfere with this metabolic network, both in a positive or in a negative way, in order to try to strengthen the resilience and prevent, basically, what is strengthening the resilience is preventing the outgrowth of certain
maturation, right?
Maturation to a certain level. Indeed. So there are options there also for interaction, for sure.
I think one factor, the final factor that we want to mention, by the way, this is all based on also a topic that we explored in a recent white paper that we published, so that's why we go in this certain direction, of course, but we have the factors of the biofilm, we have the factor of the immune function, but there's one very important factor that's also influencing this whole topic of resilience, and that is the saliva. The importance of saliva. So maybe you could elaborate a little bit on what is the role of saliva and how can it support resilience both in a healthy and in a diseased dysbiosis state.
Well, the saliva is often they say that it's a mirror of systemic health.
Yeah.
So a lot of the saliva is being more and more used as a diagnostic marker for several systemic diseases. So it's actually a very active product which is composed of a lot of metabolites that come in close contact with your biofilms. So saliva has an important, for instance, buffering capacity. It's an important mediator to regulate inflammation. So if your saliva is buffering well, your biofilm is most of the time, from a tooth decay perspective, in eubiotic state.
Yeah.
What you clearly see is if you have patients where, for instance, the salivary flow is reduced, like xerostomia, then you see that these biofilms go way easier to a dysbiotic state. So that actually shows that your saliva amount, but also probably quality, is very important in keeping your eubiotic state of your biofilm.
That makes sense, yeah.
So same goes with in diabetic patients. We know that these diabetic patients are more susceptible to certain oral diseases, which relate on one hand obviously to systemic inflammation, but also to changes in salivary composition.
We have discussed about the oral microbiome. You talked about one of its ability, so the resilience. However, I would like to tackle one of your core interests, one of your research interests, which is actually what you said, the modulation of the oral microbiome. Can you tell us why would you be interested in modulating oral microbiome, and what's the goal?
For me, I find oral biofilm modulation this is my baby. This is one of the coolest thing that exists here. And it comes a little bit from the aspect that as a clinician, I realized that I could not sterilize the oral cavity. I understood that actually the biofilm which is there has a really protective role, that we have to start thinking alternative ways, because obviously everybody knows about the antibiotic resistance, but we start to realize, and the data are getting really, really clear, there is also something which is called antiseptic resistance that is developing. So there should come new ways in order to modulate, and especially with the whole ecological hypothesis, it became clear to me that we have to balance more and more these biofilms, because they are protective on their own. I have enough patients, again, I already took the example, that are 60, 70 years old, where the oral hygiene is bad and they don't have any disease, and it's actually very protective. So a little bit by coincidence, I ended up in the probiotic research. The initial research that I did was on microbial interactions, the crosstalk. What could they do? It easily went into what is now being translated as probiotics work. Then you start that, okay, what am I doing? Modulating biofilms. So you start to look further, say, but there's also these prebiotics concept, the postbiotics, the symbiotics. Then you find predatory bacteria. Then you find stories about people who do fecal microbiome transplantations. So you start to imagine that maybe people will do oral microbiome transplantation in the future. Yeah. And suddenly you're surrounded with all sorts of treatments that are all focusing on keeping the oral microbiomes in check, keeping them in balance, and bringing dysbiotic states back to symbiotic states, or to eubiotic states, without really focusing on are there still pathogens or not. Again, I don't care about the fact that there are still pathogens or not. I care about what is the balance. Are there more beneficial species than pathogens? And does it influence inflammation? Is there a low grade of inflammation? And as long as I have patients that have a lot of plaque with low amounts of inflammation, and I even was saying to my students 2 days ago that I don't care that the oral hygiene of these patients is not optimal. there is no inflammation. Everything is in balance. The oral microbiome is happy. The host is happy. So why should we change this? It's fine.
So you're approaching the microbiome more from a health point of view than focusing on the disease point of view, which circles nicely back to what we mentioned in the beginning, of course, with the whole paradigm shift. It's
it is not focusing on the pathogens. It is stabilizing if the situation is a healthy situation, it is stabilizing the oral microbiome, making sure that if there is a factor that could push towards dysbiosis, that the microbiome has enough resilience to withstand that push.
Yeah, the challenge. Yeah.
So this could be that even sometimes a patient says, okay, I have a more stressful period in my life, then we have to make sure that this is a trigger to go into dysbiosis very clearly, that the microbiome can withstand that trigger and that it's resilient enough to, for a few days or a few weeks, can withstand the trigger to go into dysbiosis. But this is something that is emerging. It's not that easy. And it's trying to figure out, okay, which are the factors where we can work on with this patient and how can we strengthen this. And there the most developed domains are at the moment, for sure, the probiotics. The prebiotics is an emerging domain, but also there we are getting more and more positive results. And that is the way how, but also even with certain antiseptics, I have to be honest, I'm not completely against antiseptics, but there are antiseptics where we have shown in our own group that they are actually pretty well in preventing a dysbiotic shift, rather than just being used to kill. They actually can prevent these shifts.
But it's also a matter of using the antiseptics in a strategic way, because we cannot just say no antiseptics anymore, right?
Absolutely.
It really depends on the indications.
You really have to know the indication and what to use when. The problem is there are no guidelines around it. It's extremely difficult. It's an extremely difficult field also. So finding the right antiseptic for the right indication, there are some things, I mean, after surgery, after tooth extraction, obviously that's the very clear indications when to use an antiseptic boundaries. But when we go into daily healthcare prevention, then it becomes already a way more difficult story. So honestly, for me, in my own practice with patients, I really play with the three factors, which are the probiotics, the antimicrobials, although I use the antimicrobials there in order to improve the effect of the probiotics and the prebiotics.
Maybe we can come back to these supplements that you just mentioned, probiotics, prebiotics. But maybe can you just start with the basics, redefining first the probiotics for our audience, their mode of actions, what they do?
A lot of these terms that end up on the word biotics, and you have probiotics, prebiotics, postbiotics, and symbiotics. A whole bunch of things that are actually very difficult from each other, and you really need to understand what are the differences in order to understand what products can do and which products could deliver what they promise. So the best known word is the word probiotics. And when we talk about probiotics, we are talking about products that contain living bacteria with beneficial effects on general health, oral health. Now, beneficial is a little bit double in the sense that they can be there in a dormant state. But still, if they are rehydrated, they will become metabolically active and they are alive. This is in complete contradictory or opposite of something which we call postbiotics. So postbiotics are products that also contain bacteria, but they are in a dead state. And these are products that you could ask yourself, but why would you give a patient dead bacteria doesn't make any sense. Well, there are two reasons for that. One of them is that the fact that these dead bacteria can actually, by the products that are in the product, with their cell wall composition, they can already stimulate the inflammatory responses in a certain direction. That's one thing. Second thing, while they are preparing these postbiotics, they are often producing all sorts of antimicrobials, which can be in the product, in the postbiotic product. It's maintained in there. The big advantage for companies with working with postbiotics is that in terms of stability, in terms of production, it becomes way easier because you can include a postbiotic in a toothpaste or in a mouth rinse, which is an easy format that the patient understands and is used to using.
And that's something that's not possible with the pro?
It's not possible with the current probiotics. Or unless you're going to use very fancy techniques and then you're going to have a toothpaste which costs about 100 euros for one toothpaste, which is impossible. So basically all the products which are on the market that are a toothpaste and that claim that they're a probiotic toothpaste, unless the toothpaste is yogurt and has a shelf life of 2 weeks, it will be a postbiotic, which is fine. I mean, it doesn't matter. It's just another concept, another way of interacting. If we are really talking about probiotic supplements, probiotic products, it is almost all of the time in a tablet form, in a powder form, something which needs to be rehydrated. Basically, probiotics work on the two parts of oral disease. Can work on the oral microbial, that's one thing, and on the host is the second part. So in terms of what effects have been shown on the oral microbiome is that these probiotics can actively produce antimicrobials. I mean, don't forget that current penicillin was actually something which was produced by a fungus.
Right.
Yeah? So for these probiotic bacteria, they produce all sorts of antimicrobial substances that can actually kill bacteria, pathogenic bacteria that are living around them. That's one thing. On the other hand, they can interfere with the colonization of other pathogens. They can influence virulence, gene expression of other pathogens. So all the aspects of which are taking place in a biofilm are influenced or can be influenced by these probiotic bacteria. It is not that one probiotic strain will do all of these effects. It's really strain dependent what they're doing, but they clearly can interfere with the microbiome. That is very clear.
Maybe for the audience who do not understand the whole species, genus type of differentiation of bacteria, what exactly how would you explain a strain-specific activity? Like, for example, the ones that you used in your research.
Yeah, so strains, if we look at bacteria, obviously they have names.
Yeah.
So we are talking about Streptococcus mutans, Porphyromonas gingivalis.
Right.
So this Streptococcus or Porphyromonas is the genus, and mutans or gingivalis is the species. But it is a little bit like the genus would be we're all human, and the species we are man. But Martijn, you and I, we are completely different men.
Yes.
Yeah, we don't look the same. So
we should ask Rachel, but I think she will agree.
We don't go there.
Okay.
I don't want to go there. So, and this is a strain difference. So the individuality is the strain level.
Yeah.
And so within the Porphyromonas gingivalis group or within the Streptococcus mutans group, you have different strains. They all behave completely different.
Okay.
They are as different as you and me. This is also reflected in probiotics. So it's very nicely shown in the gastrointestinal field that if you take, for instance, a Lactobacillus plantarum probiotic and you look at its effect on irritable bowel syndrome, that one strain can be very effective in reducing diarrhea, in reducing abdominal pain, while if you give the other strain, it's also Lactobacillus plantarum, it actually increases abdominal pains. It increases stomach ache and diarrhea. So selecting the right strain up to a strain level is extremely difficult, and it's often forgotten in literature and in daily practice.
But then, referring to what we are just saying and appealing to your clinician activity, this difference of activities of strains, does it relate somehow, or can you explain how that could maybe impact the EFP position on probiotics? Because the EFP mentioned, or in the recent guidelines, they said that the probiotics are not necessarily recommended because it might not be have an additional effect as an adjunct to subgingival instrumentation, and they don't even know if there is any effect in controlling the inflammation. But maybe you can give us your input on that, considering what you just said about the different activities of strains.
Because I think this is a major flaw in the EFP guideline. That is what they actually did, and it's a very logical step, but it shows that you don't really understand how probiotics work. They actually took all the literature they had on probiotics, because it was called a probiotic, and they just threw it on one pile and they did some meta-analysis, and they looked at the effect, and they saw there is no statistically significant effect. That is what they did. Now, I was just explaining to you guys, look, there are differences. It's not because something which is called a probiotic that it's actually effective for all of the diseases. So the big problem with the EFP meta-analysis, which is the basis of this guideline, is that they combine probiotic, I'm not even going to say strains, species that we know that are not effective for periodontal disease. They can be effective for tooth decay, but not for periodontal disease. And they combine it with species that are effective for periodontal disease. So if you would separate those studies that use species where we know that they're effective for periodontal disease, then you see that there is a highly significant effect.
I see, yeah.
But they just threw it together. And actually, if you go to the original meta-analysis of Nikos Donos, where that they used to make this consensus statement, then actually Nikos wrote down that he saw for certain probiotic species.
I remember that sentence, yes.
Clearly that there was a better performance trend.
Yeah.
Now, this makes that we are facing actually a very strange situation within the EFP, because what you might not know is that actually in the recent EFP clinical practice guidelines for peri-implant mucositis, they are actually saying that the use of probiotics can help in the prevention and in the treatment of peri-implant mucositis.
Was that because you were part of a meta-analysis this time or not?
Well, actually, no. Yeah. So in both in both consensus meetings, I was not involved in the analysis of this data, which is logical. In the one where they voted against probiotics, I was present. In the ones where I was not present, they voted in favor of the probiotics. Now, what is the difference between the both things? It is that in peri-implant mucositis, by coincidence, all of the clinical trials were done with one probiotic strain and species where we know that from a periodontal point of view,
it is effective.
So it's a pure coincidence.
Right.
All of these studies were done with that probiotic strain.
Right.
And then you clearly can see
that should have been a sub-analysis of probably the meta-analysis in the EFP guideline was done technically perfect.
Yeah.
But it was flawed from the beginning because they threw everything on one pile, whereas they should have started with pulling data apart.
I'm very critical, so I'm not happy with the EFP practice guideline about we do not recommend to use probiotics as an adjunct to non-surgical periodontal therapy. But I'm also not completely happy with the guideline that, okay, we suggest to use probiotics, because what they should say is they should specify the species, at least the species. So it's not now that it's like now they say, okay, you can take whatever probiotic and it will help against perioperative mucositis. No way. It will depend on the species. It will probably depend on the strain. It might even depend on how the manufacturer makes the product. And it even more and more data seems to indicate that it might even depend on the patient. That not every patient responds in the same way to the same probiotic.
A lot of work ahead for the revision that will happen in a few years, probably.
It will have, it will be another fight. But I mean, for me, what is at the moment the most important thing is that by that time that there are way more clinical trials that can support or disapprove the effect, so we can have a better idea, better view on what's working, what's not working, and also especially how should we work with these things, because it's not like popping an antibiotic tablet. A probiotic is more complex in terms of its use.
Maybe you can detail that, because it looks like indeed on probiotics we have a lot of studies, a lot of research that has already been done. One of the most famous supplements probably. But you said prebiotics may work differently. So can you detail that?
So what are prebiotics? Prebiotics are actually substrates that so it are not living bacteria. It's could treat as food for bacteria that is being given to an ecology in order to stimulate the growth and the metabolic activity of beneficial bacteria that are already present in that ecology. So it's a little bit like giving a fertilizer to grass. I think that's the best way I've heard to describe prebiotics. And in a way, it's a super interesting concept because it takes away one of the downsides or the difficulties of probiotics. One of the difficulties of probiotics is getting them into the oral cavity and letting them colonize there. And we know that colonization is not permanent with probiotics. With prebiotics, you actually have already the beneficial bacteria being part of that ecology, so they're always there, just stimulating their growth, or you're stimulating their metabolic activity. So that really differentiates probiotics from prebiotics. Now, finding prebiotics that interact with the microbiome is, in my opinion, way more difficult than probiotics. Because probiotics, you can throw in whatever you want. With prebiotics, you have to be really careful that you're not stimulating wrong pathways.
The bad ones and the bad ones as well, maybe.
And the bad ones as well. Because, I mean, now I'm going to say something very controversial. But if you want to prevent periodontal disease, the best prebiotic would be to give your patient high doses of sucrose. Because the high doses of sucrose will acidify the environment. The periodontal pathogens will not like that and they will not grow. They will be really unchecked. But what you get in return...
Rampant carriers, there you go.
Rampant carriers. So this is, I mean, don't take now sucrose, don't tell me, Wim Teughels has said that sucrose is a prebiotic, don't use it. But it's the concept which is there. So finding the right prebiotic supplements is it's tricky and difficult.
Maybe then do you have another example than sucrose, just to...
I've been doing research myself on prebiotics, but often these newer molecules are quite expensive. So the ones which are on the market at the moment and which are affordable and seem to work are, for instance, nitrate. This is one of the very emerging prebiotic. And then we have the allergenin of some commercial companies that really seem to... They primarily work on buffering pHs. That is their major, especially the allergenin. When we go to nitrate, for nitrate, the story is a little bit different in the sense that it goes into stimulating nitrate-reducing bacteria that are associated with health, that can also help in buffering. But the super nice part of nitrate is that the nitrate reducing bacteria, the beneficial bacteria, are actually involved in something which is called the nitrite nitric oxide pathway. And that becomes really important because the end product of this pathway is nitric oxide, which is extremely important for general health. So it controls blood pressure, it's important in wound healing, and so on and so on. So what am I telling here is that the oral microbiome and the nitrate reducers in the oral cavity are actually connected to systemic health. And by promoting them with a prebiotic, with a nitrate, you actually could not only improve the oral health, but you would also be able to improve systemic health, general health, preventive. So not only by when there is disease, you try to improve oral health because we know about oral health, systemic health link, but actually by stimulating the oral microbiome, specific parts of that oral microbiome, you actually could create a eubiotic state, but not only for orally, but also
systemic benefit. And this nitrate, nitrite, nitric oxide pathway, it's only possible because of the oral the oral bacteria. That's incredible.
It's an incredible system. I don't know why anybody didn't think or realize this earlier, but actually the system is that this is the breakdown pathway. So you have nitrate, which you get from the green vegetables.
Yeah.
When Martijn, when your mother said, Martijn, eat your green vegetables, she was really right. It's important. Nitrate is being converted to nitrite. That goes through the stomach, but the nitrite cannot be converted to so nitrite to nitric oxide is not possible. So what it's being done is that it's being recycled through the kidneys, and it goes back to the saliva, and there the nitrate is being exposed to the oral bacteria, the nitrate reducers, who bring it to nitrite. And nitrite is then later on taken up again into the stomach and comes into systemic circulation and becomes reduced to nitric oxide with all of its benefits. But there are no cells in the human body that can do this. The only place where this happens is in the oral cavity. And that is, for instance, this is shown in several studies where when you use broad spectrum antimicrobials that actually bluntly kill all of the bacteria in the oral cavity, you could see that these patients have increased blood pressures. Also, don't see normally if you have been doing some workout, you have a post-workout hypotension, so your blood pressure go down. This is not happening with patients that use very broad, very aggressive antiseptics. And we also know from a Mexican survey or Mexican study that people who frequently use antiseptic mouth rinses, and this was determined to be 3 times a day or more, they actually have higher risks to develop diabetes and several other systemic diseases that are related to reduced nitric oxides situations.
Incredibly fascinating.
So the link of these nitrate reducers and with systemic health and the oral cavity is extremely important.
Yeah. I had one last point on the prebiotics topic. You mentioned that for the postbiotics, usually when you see toothpaste or a mouthwash that claims it's a probiotic, it's actually postbiotic because probiotics cannot be delivered in these type of products. How is that with prebiotics? What is usually the mode of delivery? Well, you already mentioned nitrates being consumed through vegetables, but how does it work for more the, I would say, artificially produced prebiotics?
I think, if we think about oral health, these are typical products that can be delivered in toothpaste amount. That would be a very logical mode of application.
So it's topical application.
Topical. This is the way I would go, unless we really can have proof that the GI microbiome is really influencing the oral microbiome. But then we need to go for completely different prebiotics. So, for instance, for the GI microbiome, the gastrointestinal microbiome, the prebiotics are often fiber prebiotics.
Yeah.
And this Gauss and Foss. The issue is there that it takes the microbiome a long time to really digest them and cut it down in different pieces. This is there, the oral microbiome is different because the passing time is really short. So you need to have other prebiotics that are fastly metabolized or taken on in your biofilm or biobacteria. So therefore, a topical local application of a prebiotic is the way to go, not the typical prebiotics for GI health.
But it seems like, prebiotics have their advantages, like probiotics, but somehow probably you can combine them into what we heard as being co-symbiotics. But there are these combinations, but maybe also with other micronutrients, or what's your take on that?
The symbiotic is one of, again, these biotic ticks, and it means that you combine a probiotic and a prebiotic. Now, the combination can be done for two reasons. So one reason can be that the prebiotic is stimulating the probiotic, so it's specifically there for the probiotic. Another reason can be that you just have a probiotic and you have a prebiotic which do not interact with each other, but you use them for another. Sure. So these are the differences. Next to that, there are a whole bunch of micronutrients where we in literature have some evidence that they actually can help in modulating microbiomes, but also most of the time steering inflammation. We should not forget that, especially when we're talking about periodontitis, periodontitis is actually a group name of, at least that's how I look at it, of different diseases that have the same clinical image, bone loss around teeth. But it's not one disease entity. I mean, it can be that for one patient, it can be because the patient has a low for vitamin D or vitamin C score. That is something which is very well known, and I also see this in my patients. So often when I see, I'm not going to say the old, the 60, 65-year-old patient who comes in with severe periodontitis, okay, that's something else. But if the 35, 40-year-old person comes in with periodontitis, I actually do a blood screen, and I screen for the micronutrients, for vitamins and the presence. And often you see, especially in our countries when there is not a lot of sun, especially wintertime, you see that the vitamin D depletion is often there. And we have a lot of evidence that shows that vitamin D is really important in strengthening the immune response.
Probably works through the immune system.
It works through the immune system. So combining these supplements together with prebiotics is for me a very logical step, because we cannot, based on the clinical picture we get from periodontitis, it's extremely difficult to find out what is now the etiological component is in this patient. Is it a depletion in micronutrients? Is it an hyper-responsive inflammatory response? Is it a hypo-inflammatory influence? Is it a certain microbial perturbation? Even viral infections have been described in the literature. So it's very, very complex. So by supplementing with these products with micronutrients, you could, based on the literature, you could improve their effectiveness. For me, this is a very logical step and a very logical combination.
But still, for the moment, a budding area of research, like something still in development, not necessarily.
Oh, yes, Rachel. I have to admit that what I'm talking now about is really something which is emerging over, for the big public over the past 5 years. I know I'm working on probiotics for 25 years now. We were pioneering that. But now, in the meantime, the prebiotics, the micronutrients are coming. And people start indeed to understand that it's this ecological balance, and that the way how we were working in the past, that's not going to work anymore. Yeah. So it's under development, but I'm quite sure that within the next 5 to 10 years, you will see way more and more these products popping up. these approaches coming, and the only thing what I'm hoping is that the products that will pop up and the approaches that are coming, that these are backed with science. Because one of the big problems that these pro-microbial therapies are suffering from is that there is this connotation that this is based on pseudoscience, which is not the case at all. Many of the claims are really based on
Actual research.
evidence, but you need to be willing to look for it, to understand it, and then you will see that it's not some hocus pocus.
And this whole field of pro and prebiotics, it's what's happening now. Maybe as a final question, how do you see the future?
What I see, there is a difference between what I see and what I hope, but obviously I think what I see for sure is that we are going to go for sure in the direction of microbiome modulation and host modulation. Way more as a preventive measure and as a treatment measure, way more than just killing bacteria. I see that in the future, we probably as dentists or healthcare workers will not be able to use any antibiotics anymore. And I feel even that they're going to restrict certain antiseptics. So we're going to be blocked there. Where I envision this is going is this will be going, and I know people are using this word already from when I was still a dental student, personalized medicine. This is one of these words that we love to use back in the 2010s, but nothing really happened. We are still treating our patients with the same techniques, which are effective for most of them, but for certain not. Now, what I see, at least in the probiotic field, I have to be careful with the prebiotic field, is that we see that these probiotics can also be host specific. So it means that if we three here would be using a probiotic, it might be that it is effective in Rachel, it's effective in you, Martijn, but they are not colonizing my oral cavity. So in a way, I think that in the future, we might find ways to figure out which probiotics are more for your health, which probiotics are more indicated for my health, not only for what they're doing, but also in terms of compatibility with my natural, and which ones are for you, Richard. So that is where I think we are going.
Sounds like taking the paradigm shift a step further and more also considering the whole host relationship.
It is, because it's for sure, I mean, whenever I'm talking about oral health, in my mind, it's immediately connected to systemic health. Oral health and systemic health are, for me, one and the same. Obviously, I'm a dentist, I'm a periodontologist, so I'm more relaxed in talking about oral health. But by improving this, by improving the resilience, by improving the microbial balances in the oral cavity, if I see what momentum we are in now and how research groups are starting to focus more and more on these ecosystems, it will not take a long time before our friends of the gastrointestinal field will be looking at the oral microbiome to get ideas. So people ask me, what is happening in the GI field in terms of, but actually the big advantage that we have in the oral field is that we can easily sample. We can learn, it's so reachable. And this is what often the gastroenterologists envy us. Because for them to take a sample,
we all know where that's going.
Okay. I don't have to explain that. Good. No, but it's true.
Yeah.
We have a very fascinating environment.
Yeah. And patients who are regularly seen by professionals as well.
And I think it's more fun to be seen by a dentist than by a gastroenterologist. I mean, many of our practitioners are convinced of that. That is not something new. I think the big challenge will be the opposite for the general practitioners, the gastroenterologists, the medical doctors, to convince them about how important the oral cavity is for their own specialty. I think that is the challenge for the coming years.
Nice. Yeah. I think that's a nice closing remark, right?
On this note, I think we can come to a close to our conversation with you, Wim. Thank you so much for this exchange and for sharing your knowledge and insights with us and with our audience. Yeah. It's been a real pleasure. Thank you all for tuning in and for being with us with this episode on the oral microbiome with Professor Wim Teughels. We hope that you could leverage some of the insights shared with us by our expert and that you can use that in your daily practice. We will be back soon with a next episode with a new guest and a new topic. Until then, thank you so much for being with us and see you soon.